Hello there, and welcome to Whispers of Science, where groundbreaking discoveries get soft spoken and curiosity curls up with cutting edge research. Tonight, we're embarking on a gentle journey into one of the most revolutionary tools in modern biology, something that sounds like it belongs in a bowl of cereal, but is actually rewriting the very code of life itself. CRISPR, the gene editing technology that's quietly transforming medicine, agriculture, and perhaps the future of humanity. Before we dive into this molecular marvel, I'd love to know where in the world you're listening from tonight and what draws you to the intersection of science and wonder. It's fascinating, isn't it, that here we all are scattered across time zones, each arriving at this moment with our own curiosity about how life works at its most fundamental level. If you find this voyage soothing, intriguing, or just a nice way to unwind after pondering the mysteries of existence, I'd be ever so grateful if you gave the video a like and perhaps subscribed it. Helps this quiet corner of discovery reach others who might need a peaceful pause with their science. Now, I do hope this exploration of genetic engineering isn't so captivating that it keeps you awake. But if it does well, at least you'll have learned something about the tools that might one day cure diseases before you drift off to dream of molecular scissors and cellular repair. So settle in, slow your breathing, and let's begin. Because somewhere between laboratory benches and living cells, between ancient bacteria and future medicine, lies a story that's reshaping what it means to edit the blueprint of life. Imagine for a moment that you could open the instruction manual for any living thing, a manual written in a language so precise that changing just one letter could transform blue eyes to brown, cure a genetic disease, or make a plant resistant to drought for most of human history. This manual, the genetic code, has been locked away. Readable, perhaps, but certainly not editable. We could observe the typos but never correct them. We could see the flaws but never fix them until now. CRISPR, which stands for clustered, regularly interspaced, short palindromic repeats, though don't worry about memorizing that mouthful, has given us something we've never had before, a molecular word processor for DNA. It's elegant, it's precise, and it's surprisingly simple in concept, though magnificently complex in its implications. Think of it as the ultimate find and replace function, except instead of fixing typos in a document, you're correcting errors in the code that makes you. But how did we discover this remarkable tool? The story begins not in a gleaming laboratory or a university research center, but in the most unlikely of places, the ancient evolutionary arms race between bacteria and viruses. You see, bacteria, those tiny single celled organisms that have been around for billions of years, have been dealing with viral infections long before humans even existed, and like any organism, under attack, they developed defenses. Viruses are essentially genetic pirates. They hijack bacterial cells, forcing them to produce more viruses until the cell bursts, like a biological piñata spilling new viruses everywhere. This is obviously bad news for the bacteria. So, over millions of years, they evolved a sophisticated immune system that could recognize and destroy viral DNA when it showed up uninvited. This bacterial immune system is what we now call CRISPR. The way it works is rather like having a molecular bouncer at the cellular nightclub bacteria. Keep a genetic mugshot book of troublesome viruses they've encountered before. These mugshots are stored as short DNA sequences in their genome. When a virus tries to break in the bacteria, can quickly flip through their mugshot book, and if they find a match, they deploy molecular scissors to cut up the viral DNA before it can cause trouble. It's remarkably efficient and surprisingly specific. But here's where human ingenuity enters the story. Scientists, particularly Dr. Jennifer Doudna and Dr. Emmanuel Charpentier, who shared the Nobel Prize for this work, realized that this bacterial defense system could be repurposed. They asked a simple but profound question: what if we could program these molecular scissors to cut any DNA sequence we wanted, not just viral DNA, but any genetic sequence in any organism? The breakthrough came when they figured out how to create a guide, RNA, a kind of molecular GPS that could direct the CRISPR scissors to any specific location in a genome. It's like giving the bouncer a new photo to look for, except now, instead of just looking for troublesome viruses, the bouncer can be programmed to find any genetic sequence. You specify a mutation that causes disease, a gene that makes a plant vulnerable to pests, or even a sequence that could be improved or enhanced. What makes CRISPR so revolutionary is its combination of precision, speed, and accessibility. Before CRISPR, gene editing was possible, but it was slow, expensive, and often imprecise, like trying to edit a book by randomly rearranging letters and hoping for the best. CRISPR changed all that. It made gene editing fast, affordable, and remarkably precise. Suddenly, laboratories around the world could edit genes with the same ease that you might edit a document on your computer. The implications began to unfold almost immediately in medicine. Researchers started exploring how CRISPR could treat genetic diseases that had been considered incurable: sickle cell anemia, a painful condition caused by a single letter change in the genetic code, suddenly became a target for potential cure. Huntington's disease, muscular dystrophy, even certain types of blindness, all conditions written into someone's DNA from birth, could theoretically be edited out in agriculture. Scientists began developing crops that could withstand droughts, resist pests, or provide better nutrition: wheat that could grow in salty soil, rice enhanced with vitamin A to combat malnutrition, corn that could resist devastating plant diseases. The possibilities seemed to multiply with each passing month. But perhaps most intriguingly, CRISPR opened up possibilities we'd only dreamed of in science fiction: the ability to edit human embryos to prevent genetic diseases from being passed to future generations, the potential to enhance human capabilities, or even to bring back extinct species by editing their genetic code into related living organisms. Now I know what you might be thinking. This all sounds rather like playing God with the fundamental building blocks of life, and you wouldn't be wrong to have that thought. CRISPR raises profound ethical questions that we're still grappling with as a society. When is it appropriate to edit human genes? Should we enhance humans beyond their natural capabilities? What are the risks of unintended consequences? And who gets to decide how this powerful technology is used? These aren't just academic questions, they're pressing concerns that affect all of us, because CRISPR doesn't just promise to treat the sick, it promises to redefine what normal means for human beings. It could eliminate genetic diseases, but it could also create genetic advantages available only to those who can afford them. It could solve agricultural challenges, but it could also create new dependencies and risks in our food system. The technical elegance of CRISPR is matched only by the complexity of the questions it raises, and perhaps that's fitting for a technology born from the ancient battle between bacteria and viruses. Evolution has always been about adaptation, survival, and change. CRISPR simply gives us the power to direct that change consciously, deliberately, with intention, rather than leaving it to the slow, random processes of natural selection. What's particularly fascinating is how CRISPR works at the molecular level. The system has three main components: the guide RNA, which acts as the GPS directing the scissors to the right location; the Cas protein, usually Cas, which acts as the molecular scissors, cutting the DNA at precisely the right spot; and the DNA template, which can be used to repair or replace the cut section with a desired sequence. When these components are delivered into a cell, they work together like a highly trained surgical team. The guide RNA scans the genome, looking for its target sequence. When it finds a match, it signals to the Cas protein, which then makes a precise cut in the DNA. The cell's natural repair mechanisms kick in, and if you've provided a template, they'll use it to fix the break, inserting your desired genetic change. But the beauty of CRISPR lies not just in its precision, but in its programmability. Unlike previous gene editing tools, which were designed for specific targets, CRISPR can be reprogrammed simply by changing the guide RNA. It's like having a universal key that can be recut to open any lock you need. This flexibility has accelerated genetic research at an unprecedented pace. Laboratories that once spent months or years developing tools to edit a single gene can now edit multiple genes in a matter of days or weeks. Experiments that were once prohibitively expensive are now accessible to researchers around the world. This democratization of gene editing has led to an explosion of research and discovery. In just over a decade since its development, CRISPR has already shown remarkable promise in treating human diseases. The first CRISPR therapy was approved for clinical use in treating sickle cell disease patients who once faced a lifetime of pain and complications, now have hope for a cure through editing their own cells to produce healthy hemoglobin. Researchers are also exploring CRISPR's potential in treating cancer by editing immune cells to better recognize and attack tumors, in combating viral infections, including HIV, by cutting the virus out of infected cells, and in addressing age related diseases by repairing the genetic damage that accumulates over time. But CRISPR's impact extends far beyond medicine. In agriculture, it's being used to develop crops that can adapt to climate change, providing food security in an uncertain world. Scientists have created rice that can survive flooding wheat, that can grow in drought conditions, and fruits that last longer without spoiling. Environmental applications are equally promising. CRISPR could help save endangered species by correcting genetic problems in small populations, control invasive species by introducing genetic changes that limit their reproduction, or even help ecosystems adapt to environmental changes more quickly. Yet with each new application comes new questions and considerations. The power to edit life brings with it the responsibility to use that power wisely, and that wisdom requires not just scientific knowledge, but also ethical reflection, social dialogue, and careful consideration of consequences, both intended and unintended. As we continue our exploration of CRISPR in the coming segments, we'll delve deeper into how this remarkable technology actually works at the molecular level, examine the most promising applications being developed today, and grapple with the profound questions it raises about the future of life itself because CRISPR isn't just a scientific breakthrough. It's a mirror that reflects our deepest values, our greatest hopes, and our most fundamental questions about what it means to be human in an age where we can rewrite the code of life. The whispers of science are telling us that we stand at a threshold unlike any in human history, where the boundary between what is and what could be has become beautifully blurry and wonderfully uncertain. To truly understand how CRISPR works, its molecular magic, we need to journey deep into the cellular world where DNA lives and breathes, where the very essence of life is written in four simple letters A, T, G, and C, arranged in sequences that stretch for billions of characters, like the longest book ever written. Except this book doesn't just tell a story, it builds and maintains every living thing on Earth. Picture, if you will, a vast library contained within every cell of your body. This library holds roughly 3 billion books, each book representing a single nucleotide, and together they contain the complete instructions for making you from the color of your eyes, to the way your immune system responds to threats, to the proteins that help you digest your breakfast this morning. Every function, every trait, every possibility is encoded somewhere in this vast molecular library. Now imagine that somewhere in this library there's a typo, a single letter that's wrong perhaps. Instead of the letter A, there's a T, or where there should be a G, someone wrote a C. It seems like such a small error in such a vast collection. But this tiny mistake could mean the difference between healthy blood cells and sickle-shaped ones, between clear vision and blindness, between a long, healthy life and a devastating genetic disease. For most of human history, we could read these books, we could identify the typos, but we couldn't fix them. We were like librarians with access to the world's most important collection, but no eraser, no correction fluid, no way to fix the errors. We found that changed when scientists learned to harness the power of a system that bacteria had been using for millions of years to protect themselves from viral attacks. The CRISPR system, in its natural bacterial habitat, works rather like a sophisticated security system. Imagine a building with the most advanced surveillance technology. Every time an intruder tries to break in the security cameras, capture their image, and store it in a database. If that same intruder ever tries to break in again, the system recognizes them immediately and dispatches security guards to remove them before they can cause any damage. Bacteria face constant attacks from viruses called bacteriophages, which are essentially genetic pirates that hijack bacterial cells to make copies of themselves over millions of years. Bacteria evolved CRISPR as their security system. When a virus injects its DNA into a bacterial cell, the CRISPR system captures a small piece of that viral DNA and stores it like a mugshot in a special part of the bacterial genome. If the same virus ever attacks again, the CRISPR system recognizes the invader by comparing its DNA to the stored mugshots. When it finds a match, it creates a guide RNA that's perfectly complementary to the viral DNA sequence. This guide RNA then recruits a protein called Cas9, which acts like molecular scissors together. They hunt down the viral DNA and cut it to pieces, destroying the threat before it can replicate. What scientists realized was that this natural system could be reprogrammed. The guide RNA, which normally looks for viral DNA, could be designed to look for any DNA sequence they wanted, and the Cas9 protein would dutifully cut whatever the guide RNA pointed it toward. It was like discovering that you could reprogram a security system to look for any face you wanted, not just known criminals. The elegance of this system lies in its simplicity and specificity. The guide RNA is typically about 20 nucleotides long, which means it can distinguish its target from every other sequence in a genome with extraordinary precision. To put this in perspective, imagine trying to find one specific sentence in a library containing millions of books. The guide RNA can do exactly that, scanning through billions of DNA letters to find its perfect match. When the Cas9 protein makes its cut, it creates what scientists call a double strand break in the DNA. This might sound alarming, but cells have sophisticated repair mechanisms that spring into action whenever their DNA is damaged. They can repair the break in one of two ways. They can simply glue the broken ends back together, which sometimes results in small insertions or deletions that can disable a gene, or they can use a provided template to repair the break, inserting new genetic information in the process. This repair mechanism is where the real power of CRISPR lies. By providing the cell with a specific DNA template, scientists can essentially trick the cell into making any genetic change they want. They can correct disease causing mutations, insert new genes, or even swap one gene for another. The cell's own machinery does the actual editing. CRISPR just tells it where and provides the replacement parts. The speed and precision of CRISPR have revolutionized genetic research. What once took months or years can now be accomplished in days or weeks. Experiments that were once prohibitively expensive are now accessible to researchers around the world. This democratization of gene editing has accelerated the pace of discovery in ways that would have been unimaginable just 2 decades ago. But the true marvel of CRISPR isn't just its technical capabilities, it's the window it opens into the fundamental nature of life itself. Every living thing on Earth, from the smallest bacteria to the largest whale, shares the same basic genetic code, the same 4-letter alphabet, the same fundamental mechanisms for storing and expressing genetic information. CRISPR works across all of these organisms because life at its core is remarkably unified. This universality means that discoveries made in one organism often apply to others. A gene editing technique developed in bacteria might work in plants, which might inform approaches in human medicine. The boundaries between different fields of biology have become increasingly fluid as researchers realize that we're all working on variations of the same fundamental themes. Consider the remarkable journey that led to CRISPR's discovery. It began with curiosity driven research into how bacteria defend themselves from viruses. No one was trying to develop a gene editing tool. Researchers were simply trying to understand how these ancient microorganisms survived in a world full of genetic predators. Yet this basic research into bacterial immunity led to one of the most powerful biotechnologies ever developed. This is often how science works. The most profound discoveries emerge from the most unexpected places. A curious observation about bacterial behavior leads to a Nobel Prize winning technology that could transform medicine, agriculture, and our understanding of life itself. It's a reminder that we never know where the next breakthrough will come from or how seemingly unrelated fields of study might suddenly converge. The story of CRISPR also highlights the collaborative nature of modern science, while Jennifer Doudna and Emmanuel Charpentier are often credited as the primary inventors of CRISPR. Their work built on discoveries made by dozens of other researchers. Over many years, the Spanish microbiologist Francisco Mojica first described the repetitive DNA sequences that give CRISPR its name. Dutch researcher John van der Oost helped characterize the system. French scientist Sylvain Monod contributed to understanding how it works. This collaborative effort continues today as thousands of researchers around the world work to refine, improve, and apply CRISPR technology. Each new study, each failed experiment, each successful application adds to our collective understanding and pushes the boundaries of what's possible. It's a beautiful example of how human curiosity and collaboration can unlock the secrets of nature. The technical details of how CRISPR works might seem complex, but at its heart, it's based on principles that are surprisingly intuitive. It's about recognition, finding the right target in a vast sea of possibilities. It's about precision making changes exactly where they're needed without affecting anything else. And it's about repair using the cell's own mechanisms to heal and improve itself. These principles apply not just at the molecular level, but at larger scales as well. Just as CRISPR can find and fix specific genetic sequences, researchers are learning to find and address specific problems in complex biological systems. They're developing ways to edit not just individual genes, but entire genetic pathways and even whole ecosystems. The implications ripple outward in ways we're only beginning to understand. If we can edit genes, we can potentially edit the trajectory of genetic diseases. If we can edit the genes of crops, we can potentially address food security and climate change. If we can edit the genes of other organisms, we might be able to restore damaged ecosystems or even bring back extinct species. But with each new possibility comes new responsibility. The power to edit life is not a power to be wielded lightly. It requires wisdom, humility, and a deep appreciation for the complexity and interconnectedness of biological systems. Every change, no matter how small or well intentioned, could have consequences that ripple through generations. As we stand on the threshold of this new era of genetic medicine and biological engineering, we find ourselves grappling with questions that previous generations could never have imagined. What does it mean to be human when we can edit our own genes? How do we ensure that the benefits of genetic engineering are shared equitably? How do we balance the desire to eliminate suffering with respect for natural diversity? These questions don't have easy answers, but they're questions we must engage with as we continue to develop and apply CRISPR technology. Because the choices we make today about how to use this powerful tool will shape the future of life on Earth for generations to come. The whispers of science are telling us that we've crossed a threshold, that we've gained the ability to direct evolution itself. But with that ability comes the profound responsibility to use it wisely, thoughtfully, and with deep respect for the intricate beauty of the living world we're privileged to call home. The applications of CRISPR stretch across the landscape of human experience, like ripples spreading from a stone dropped in still water, each wave carrying the potential to transform how we understand and interact with the living world. In laboratories around the globe, Researchers are quietly working on applications that would have sounded like pure fantasy just a generation ago. Yet here we are, witnessing the gentle revolution of genetic possibility. Let's begin with medicine, where CRISPR whispers the most immediate promises of healing. Imagine walking into a clinic not to manage a genetic disease, but to cure it entirely at its source sickle cell disease, that cruel condition where a single letter change in the genetic code transforms healthy red blood cells into painful, crescent shaped obstacles, has become one of the first targets for CRISPR therapy. Patients can now have their own bone marrow cells extracted, edited to produce healthy hemoglobin, and returned to their bodies like molecular transplants carrying the gift of normalcy. The process is remarkably elegant in its simplicity. Scientists remove stem cells from the patient's bone marrow, the cellular factories that produce blood cells throughout our lives. Using CRISPR, they make precise edits to correct the genetic typo that causes the disease. These corrected cells are then infused back into the patient, where they take up residence and begin producing healthy blood cells. It's like replacing a broken printer with one that works perfectly, except the printer is microscopic and the documents it produces are the very cells that carry oxygen through your body. But sickle cell disease is just the beginning. Researchers are exploring CRISPR treatments for conditions that have long been considered untouchable. Duchenne muscular dystrophy, where missing genetic instructions lead to progressive muscle weakness, could potentially be treated by inserting the correct genetic code. Certain forms of inherited blindness might be cured by editing the cells of the retina. Even some cancers, those rogue cellular societies that have forgotten how to behave properly, could be addressed by editing immune cells to better recognize and eliminate tumors. The approach to cancer treatment is particularly fascinating. Scientists can extract a patient's T cells, the immune system's special forces, and edit them to better recognize cancer cells. These enhanced immune cells, sometimes called CAR T cells, are then returned to the patient where they can hunt down cancer with unprecedented precision. It's like upgrading the security system in your body, giving your immune cells better tools to identify and eliminate threats. Moving beyond human medicine, CRISPR is quietly reshaping agriculture in ways that could help feed a growing world while adapting to a changing climate. Traditional plant breeding, which relies on selecting favorable traits over many generations, is a process measured in years or decades. CRISPR compresses this timeline dramatically, allowing scientists to make precise genetic changes in a single generation. Consider rice, the staple food for more than half the world's population. Scientists have used CRISPR to develop varieties that can survive flooding, a crucial adaptation as climate change brings more extreme weather patterns. Other researchers have created wheat that can grow in salty soils, potentially opening up new agricultural land in coastal areas. The changes are often remarkably subtle, perhaps editing just a few letters in the plant's genetic code to enhance its natural resilience. The beauty of these agricultural applications lies not in creating something entirely artificial, but in accelerating natural processes that might otherwise take decades or centuries. Plants have always adapted to environmental challenges through genetic variation and selection. CRISPR simply allows us to guide this process more deliberately, helping crops adapt to the challenges they'll face in a rapidly changing world. But perhaps even more intriguing are the environmental applications that researchers are beginning to explore. CRISPR could potentially help us address some of the ecological crises that threaten biodiversity around the world. Invasive species that wreak havoc on native ecosystems might be controlled through genetic modifications that limit their reproduction or make them less competitive. Endangered species suffering from genetic bottlenecks could have their genetic diversity enhanced, giving them a better chance of survival. The American chestnut tree, once a dominant species in eastern North American forests before being decimated by an invasive fungus, could potentially be restored through CRISPR. The scientists are working to edit the tree's genome to include genes that provide resistance to the deadly fungus, essentially giving the species the genetic tools it needs to survive in a world where the pathogen exists. Even more ambitious is the possibility of de extinction, bringing back species that have vanished from Earth. While we can't resurrect dinosaurs like in the movies, we might be able to restore more recently extinct species by editing the genomes of their close relatives. The woolly mammoth, extinct for only a few thousand years, could potentially be brought back by editing the genome of Asian elephants to include mammoth specific traits. It's a project that captures the imagination while raising profound questions about our relationship with nature and extinction. These applications showcase CRISPR's remarkable versatility, but they also highlight the careful considerations that must guide its use. Each application brings its own set of ethical questions and practical challenges. In medicine, we must balance the desire to eliminate suffering with concerns about genetic enhancement and equality of access. In agriculture, we must weigh the benefits of improved crops against concerns about genetic diversity and ecological impact. In environmental applications, we must consider whether we have the wisdom to safely manipulate entire ecosystems. The speed at which CRISPR research is advancing sometimes outpaces our ability to fully consider these implications. What once took years of careful deliberation might now happen in months or weeks. This acceleration brings both tremendous opportunities and significant responsibilities. We're essentially learning to drive while the car is still being built, navigating questions that have no historical precedent. Yet perhaps this is simply the nature of transformative technologies. They don't wait for us to fully understand their implications before they begin changing the world. Fire, agriculture, the printing press, electricity, computers, the internet. Each of these technologies transformed human society in ways that were largely unpredictable when they were first developed. CRISPR may well be the next chapter in this story of human ingenuity and adaptation. What makes CRISPR particularly remarkable is not just its technical capabilities, but its accessibility. Unlike previous biotechnologies that required massive infrastructure and investment, CRISPR can be implemented in relatively modest laboratories around the world. This democratization of genetic engineering means that innovations and applications are emerging from unexpected places, from university labs to startup companies to research institutions in developing countries. This global accessibility also means that the governance and regulation of CRISPR technology becomes more complex. Different countries and cultures may have very different views on what applications are appropriate or ethical. What one society sees as beneficial genetic enhancement, another might view as unacceptable interference with nature. These differences in perspective and regulation create a patchwork of policies that researchers and companies must navigate. The international scientific community has been working to establish guidelines and best practices for CRISPR research, but the technology's rapid development often outpaces the ability of regulatory frameworks to keep up. This creates a situation where scientists must often rely on their own ethical compass and the informal norms of the research community to guide their work. As we continue to explore the applications of CRISPR, we're also learning about its limitations and challenges. The technology, while remarkably precise, is not perfect. Off target effects, where CRISPR makes unintended cuts in the genome, remain a concern, particularly for medical applications. Delivery systems for getting CRISPR components into the right cells in the right tissues continue to be refined and improved. There are also biological challenges that CRISPR alone cannot solve. Some genetic diseases are caused not by single gene defects, but by complex interactions between multiple genes and environmental factors. Some tissues in the body are difficult to reach with current delivery methods. Some genetic changes might have unintended consequences that only become apparent over time. These limitations don't diminish CRISPR's revolutionary potential, but they do remind us that scientific progress is rarely a straight line from problem to solution. Each breakthrough reveals new questions and challenges that must be addressed. Each success opens doors to new possibilities while highlighting the complexity of biological systems. The story of CRISPR is still being written, with new chapters added almost daily as researchers around the world push the boundaries of what's possible. From the laboratory benches, where scientists carefully design guide RNAs, to the clinical trials where patients receive their first CRISPR treatments, from the greenhouses where edited plants grow to the computer models that predict ecological impacts, the quiet revolution continues. And perhaps that's the most remarkable thing about CRISPR. It's not a single breakthrough, but an ongoing process of discovery and application. Each new study, each successful treatment, each refined technique adds to our collective understanding and capability. We're not just witnessing a scientific revolution, we're participating in it, shaping it with our choices and values and dreams for the future. The whispers of science are telling us that we've entered an era where the line between what is and what could be has become beautifully, wonderfully blurred, where the ancient dream of healing the sick and feeding the hungry has found new expression in the precise language of genetic code. But with great power comes great responsibility, and nowhere is this more evident than in the ethical landscape that CRISPR has opened before us. The questions we face aren't just technical or scientific, they're deeply human, touching on our most fundamental beliefs about life, identity, and what it means to be human in an age where we can rewrite our own genetic story. The most immediate ethical considerations arise in human medicine, where the line between treatment and enhancement becomes beautifully, troublingly blurred. When we use CRISPR to correct a genetic mutation that causes cystic fibrosis, most would agree this is therapeutic, a clear medical good. But what about editing genes to enhance memory, increase muscle mass, or improve disease resistance beyond normal human ranges? Where does healing end and human enhancement begin? These questions become even more complex when we consider germline editing, changes made to sperm, eggs, or early embryos that would be passed on to future generations. Unlike somatic cell editing, which affects only the treated individual, germline changes ripple forward through time, potentially affecting people who cannot consent to these modifications. It's like signing a contract on behalf of your great great grandchildren, committing them to genetic changes they had no voice in choosing. The scientific community learned this lesson dramatically in 2018 when a Chinese researcher announced the birth of the first CRISPR edited babies. The international response was swift and largely condemnatory, not because the technology didn't work, but because the ethical frameworks for such profound interventions hadn't been established. The researcher had essentially conducted an unauthorized experiment on the human species itself, crossing a line that most scientists felt wasn't ready to be crossed. This incident highlighted the global nature of CRISPR ethics. Science doesn't respect national boundaries, and genetic modifications made in one country could theoretically spread worldwide through normal human reproduction. What one society considers an acceptable risk or benefit might be viewed very differently by another. These cultural and religious differences in perspectives on genetic modification create a complex web of considerations that no single regulatory body can address. There's also the question of access and equity that haunts every discussion of CRISPR's medical applications. If genetic therapies can eliminate hereditary diseases or enhance human capabilities, who gets access to these treatments? Will CRISPR create a world where genetic advantages are available only to those who can afford them, essentially creating biological classes based on economic status? The specter of genetic inequality, where some humans have access to edited genes that provide advantages in health, intelligence, or physical capability, raises profound questions about justice and human dignity. These concerns aren't merely theoretical. Current gene therapies can cost hundreds of thousands or even millions of dollars per treatment, while costs may decrease over time, as they have with many medical technologies. The initial availability of CRISPR treatments will likely be limited to those with substantial resources or excellent insurance coverage. This creates a moral tension between the desire to help those who can be helped now and the concern about creating or exacerbating existing inequalities. The environmental applications of CRISPR raise their own set of ethical puzzles. When we edit the genes of wild organisms or release genetically modified organisms into the environment, we're essentially conducting experiments on ecosystems that we don't fully understand. The interconnectedness of natural systems means that changing one species could have cascading effects throughout an entire ecological web. Consider the possibility of using CRISPR to control mosquito populations by making them unable to carry malaria parasites. This could save millions of human lives, particularly in developing countries where malaria remains a leading cause of death. But mosquitoes also serve as food for bats, birds, and other animals. They pollinate certain plants and play roles in ecosystems that we're still discovering. What happens if we dramatically reduce mosquito populations? What fills that ecological niche? These questions don't have easy answers, but they demand careful consideration before we release edited organisms into the wild. The concept of consent becomes particularly complex in environmental applications. Humans can choose whether to undergo genetic modification, at least in theory, but ecosystems, other species, and future generations cannot consent to the environmental changes that might result from releasing genetically modified organisms. We're making decisions that could affect the entire biosphere based on our current understanding and values, which may seem limited or misguided to future generations. There's also the question of reversibility. Some genetic changes, once made and released into wild populations, might be impossible to undo. Unlike a medical treatment that affects only one individual, environmental applications of CRISPR could create permanent changes to the genetic landscape of entire species or ecosystems. This irreversibility demands an extremely high standard of caution and evidence before proceeding. The agricultural applications of CRISPR sit somewhere between medical and environmental ethics, carrying elements of both. Edited crops could help address food security and reduce the environmental impact of agriculture, but they also raise questions about corporate control of the food supply, the rights of farmers, and the preservation of agricultural biodiversity. Traditional farming has always involved selecting and breeding crops for desired traits, but CRISPR accelerates this process dramatically and allows for changes that wouldn't be possible through conventional breeding. This power concentration in the hands of a few large corporations that own the technology and the edited seeds raises concerns about farmer autonomy and food sovereignty. If a few companies control the genetic basis of our food supply, what happens to agricultural diversity and resilience? There's also the question of labeling and consumer choice. Do people have a right to know whether their food has been genetically edited? Different societies answer this question differently, with some requiring clear labeling of genetically modified foods, while others treat edited crops as substantially equivalent to conventionally bred varieties. These differences in approach reflect deeper philosophical disagreements about the relationship between humans, technology, and nature. Religious and spiritual perspectives add another layer of complexity to CRISPR ethics. Many faith traditions have specific teachings about the sanctity of life, the role of humans in creation, and the appropriate limits of human intervention in natural processes. Some see genetic editing as a God given tool for healing and improving life, while others view it as an inappropriate attempt to play God or interfere with divine creation. These religious perspectives aren't merely academic, they influence policy decisions, research funding, and public acceptance of CRISPR applications. In some countries, religious considerations play a major role in determining which applications of genetic editing are permitted or funded. Scientists working in this field must navigate not only the technical and safety challenges of their research, but also the diverse moral and spiritual frameworks within which their work will be evaluated. The global nature of CRISPR research creates additional ethical challenges. Scientists in different countries operate under different regulatory frameworks, cultural norms, and ethical guidelines. What's prohibited in one country might be actively pursued in another. This creates the potential for ethics tourism, where researchers move to jurisdictions with more permissive regulations to conduct studies that wouldn't be allowed in their home countries. The international scientific community has worked to establish common ethical principles for CRISPR research, but enforcement remains challenging. Professional societies can establish guidelines and withdraw membership from researchers who violate ethical norms, but they cannot prevent governments or private companies from funding research that the broader community considers inappropriate. This regulatory patchwork also creates competitive pressures. If one country allows certain types of CRISPR research while others prohibit it, researchers and companies might relocate to take advantage of more permissive regulations. This could lead to a race to the bottom in ethical standards as countries compete to attract biotechnology investment by relaxing their oversight of genetic research. The pace of CRISPR development adds urgency to these ethical considerations. Traditional bioethics has had decades or centuries to develop frameworks for evaluating medical interventions, but CRISPR applications are advancing so rapidly that ethical analysis struggles to keep up. By the time we've thoroughly considered the implications of one application, researchers have already moved on to the next breakthrough. This speed creates pressure to make ethical decisions with incomplete information. We don't yet know the long term effects of many CRISPR applications, but waiting for complete data might mean missing opportunities to help people who are suffering today. The tension between caution and compassion, between safety and urgency, runs through every discussion of CRISPR ethics. Perhaps most fundamentally, CRISPR forces us to confront questions about human nature and identity that we've never had to answer before. If we can edit our genes, what makes us human? If we can enhance our capabilities beyond their natural limits, do we remain the same species? If we can direct our own evolution, what responsibility do we have to future generations and to the other species that share our planet? These aren't just philosophical puzzles, they're practical questions that will shape how we develop and apply CRISPR technology. Our answers will determine whether genetic editing becomes a tool for healing and human flourishing or a source of division and inequality. They'll influence whether we use this power to enhance human dignity or to create new forms of discrimination and exclusion. The whispers of science are telling us that we stand at a crossroads where the choices we make about genetic editing will echo through generations, shaping not just the trajectory of human health and capability, but the very meaning of what it means to be human in a world where the genetic code of life has become as editable as a document on a computer screen. The future of CRISPR stretches before us like an uncharted map, with territories we're only beginning to explore and destinations we can barely imagine. As we peer into this genetic tomorrow, we find ourselves contemplating possibilities that would have seemed like pure fantasy just a generation ago. Yet here we are, standing at the threshold of an era where the very essence of life has become malleable, editable, improvable. In research laboratories around the world, scientists are pushing the boundaries of what CRISPR can accomplish, and their discoveries whisper of applications that could reshape civilization itself. Imagine a world where genetic diseases are no longer passed from parent to child, where crops can adapt to any climate, where damaged ecosystems can be restored with precision, and where the aging process itself becomes something we can slow or perhaps even reverse. The next generation of CRISPR technologies promises even greater precision and capability. Scientists are developing new versions of the molecular scissors that can make smaller, more subtle changes to DNA, like replacing a single letter without cutting the double helix at all. These base editors and prime editors, as they're called, work more like pencil erasers and correction fluid than scissors, allowing for incredibly precise modifications with reduced risk of unintended consequences. Prime editing, in particular, represents a remarkable leap forward in genetic precision. Instead of cutting DNA and hoping the cell repairs it correctly, prime editing can insert, delete, or replace specific genetic sequences with extraordinary accuracy. It's like having a molecular word processor that can find any sentence in the genome and rewrite it exactly as desired without affecting anything else in the vast library of genetic information. These advances in precision are opening doors to treating genetic conditions that were previously considered untouchable. Single letter changes in DNA that cause diseases like Huntington's disease or certain forms of inherited blindness could potentially be corrected with surgical precision, leaving the rest of the genome completely untouched. It's genetic surgery at its finest, more precise than any scalpel that has ever existed. But perhaps even more intriguing are the possibilities that emerge when we consider CRISPR not as a single tool, but as part of a larger toolkit for understanding and manipulating biological systems. Scientists are beginning to combine CRISPR with other cutting edge technologies, artificial intelligence, nanotechnology, synthetic biology, creating hybrid approaches that could accomplish things no single technology could achieve alone. Artificial intelligence is already helping researchers design more effective guide RNAs and predict the outcomes of genetic modifications. Machine learning algorithms can analyze vast databases of genetic information to identify the best targets for CRISPR editing, predict potential side effects, and even suggest entirely new therapeutic approaches. It's like having a brilliant assistant that never sleeps, constantly analyzing genetic data and proposing new experiments. The marriage of CRISPR and nanotechnology could revolutionize how genetic modifications are delivered to specific cells and tissues in the body. Tiny nanoparticles could carry CRISPR components directly to diseased cells, like molecular delivery trucks that know exactly where to go and what to deliver. This targeted approach could make genetic therapies safer and more effective, reducing the risk of editing healthy cells while ensuring that therapeutic changes reach their intended destinations. Synthetic biology, the field that treats biological systems like engineering projects, is beginning to incorporate CRISPR as one tool among many for designing and building new biological functions. Scientists are learning to create genetic circuits that can respond to environmental conditions, produce valuable compounds, or even perform computational tasks inside living cells. CRISPR becomes part of a larger language for programming biology, allowing researchers to write genetic software that runs on cellular hardware. These technological convergences point toward a future where biological engineering becomes as precise and predictable as electronic engineering. We might eventually be able to design biological systems with the same confidence that engineers design computer chips or bridges, creating living machines that can solve problems, produce materials, or even repair themselves. The implications for medicine are staggering. Imagine therapeutic cells that could be programmed to patrol your body, detecting and eliminating cancer cells, clearing arterial plaques, or repairing damaged tissues. These biological robots could be created from your own cells, edited to perform specific functions, and returned to your body where they would work tirelessly to maintain your health. Regenerative medicine could be transformed by combining CRISPR with advances in stem cell research and tissue engineering. Scientists might be able to take a small sample of your cells, edit them to correct genetic defects, or enhance their regenerative capabilities, and then guide them to regrow damaged organs or tissues. It's personalized medicine taken to its logical extreme, where treatments are tailored not just to your disease, but to your individual genetic makeup. The aging process itself might eventually yield to CRISPR based interventions. As we learn more about the genetic factors that contribute to aging, it may become possible to edit cells to resist age related damage, maintain their youthful functions longer, or even reverse some aspects of the aging process. The fountain of youth, it turns out, might not be a magical spring, but a collection of precisely targeted genetic modifications. In agriculture, the future applications of CRISPR stretch far beyond simple crop improvement. Scientists are exploring the possibility of engineering entire agricultural ecosystems, creating crops that work together symbiotically, fixing nitrogen, sharing nutrients, and protecting each other from pests and diseases. These engineered agricultural communities could be more productive and sustainable than anything nature has produced on its own. Vertical farming systems could be revolutionized by crops specifically designed to grow in controlled indoor environments. Plants could be edited to thrive under LED lights, require minimal water, and produce maximum nutrition in minimal space. Urban agriculture could become not just feasible, but highly efficient, bringing food production closer to population centers and reducing the environmental impact of transportation. The possibility of engineering crops that can grow in extreme environments could open up entirely new agricultural frontiers. Desert farming could become viable with plants designed to thrive in arid conditions. Coastal agriculture could expand with crops that can tolerate salt water. Even space agriculture could become possible with plants engineered to grow in the unique conditions of spacecraft or planetary colonies. Environmental applications of future CRISPR technologies could help address some of the most pressing challenges facing our planet. Climate change mitigation could be supported by engineering plants and microorganisms that capture and store carbon more efficiently. Ocean acidification might be addressed by releasing edited coral species that can thrive in more acidic waters. Pollution cleanup could be accelerated by microorganisms designed to break down specific contaminants. The concept of ecosystem restoration could be revolutionized by precisely targeted genetic interventions. Instead of trying to restore damaged ecosystems by reintroducing species that may no longer be adapted to current conditions, scientists might be able to edit existing species to fill ecological niches, restore food webs, and rebuild the complex relationships that make ecosystems resilient. Biodiversity conservation could benefit from genetic rescue approaches, where endangered species receive genetic modifications that improve their survival prospects. Small populations suffering from inbreeding could have their genetic diversity enhanced. Species threatened by disease could be given resistance genes. Populations struggling to adapt to environmental changes could receive helpful genetic variants from related species. The possibility of de extinction, bringing back species that have vanished from Earth, continues to capture imaginations and raise profound questions. While we may never see living dinosaurs, more recently extinct species might be candidates for genetic resurrection. The passenger pigeon, the thylacine, even the woolly mammoth could potentially be restored through careful genetic editing of related species. But these ambitious applications also highlight the need for equally ambitious approaches to governance and ethics. As CRISPR capabilities expand, so too must our frameworks for ensuring that these powerful technologies are used wisely and responsibly. International cooperation will become increasingly important as genetic modifications cross borders and affect global commons like the atmosphere and oceans. The development of CRISPR is also driving advances in related fields that could prove equally transformative. Gene drives, genetic modifications that spread through wild populations more rapidly than normal inheritance would allow, could provide powerful tools for controlling disease vectors or invasive species. But they also carry risks that could affect entire ecosystems, requiring extraordinary caution and international coordination. Epigenetic editing, which modifies how genes are expressed without changing the underlying DNA sequence, offers another frontier for precise biological control. These modifications could potentially be reversed more easily than traditional genetic changes, providing a kind of biological volume control that could be adjusted as needed. The field of genetic diagnosis is also evolving rapidly, with new technologies that can detect genetic variations with unprecedented speed and accuracy. Portable DNA sequencing devices could make genetic testing available anywhere in the world, while artificial intelligence systems could interpret genetic data and recommend personalized interventions in real time. As we look toward this genetic future, we must also consider the social and economic transformations that widespread adoption of CRISPR technologies might bring. Traditional healthcare systems built around treating diseases might need to adapt to a world where many conditions can be prevented or cured at the genetic level. Agricultural systems might be revolutionized by crops that require entirely different approaches to farming. Environmental management might be transformed by the ability to engineer biological solutions to ecological problems. Education systems will need to prepare future generations for a world where genetic literacy becomes as important as traditional literacy. Citizens will need to understand enough about genetics and biotechnology to make informed decisions about their own genetic modifications and to participate meaningfully in democratic discussions about how these technologies should be regulated and applied. The economic implications are equally profound. New industries will emerge around genetic engineering services, while existing industries might be disrupted by genetically enhanced alternatives. The pharmaceutical industry might shift from treating diseases to preventing them through genetic modifications. Agriculture might be transformed by crops that require fewer inputs and produce higher yields. Environmental services might expand to include ecosystem engineering and genetic conservation. But perhaps the most important consideration is how we maintain our humanity in an age of genetic enhancement. As we gain the power to modify our own biology, we must thoughtfully consider what aspects of human nature we want to preserve and what limitations we're willing to accept. The goal shouldn't be to create perfect humans, but to use these tools to reduce suffering, enhance human flourishing, and protect the diversity that makes life beautiful and resilient. The whispers of science are telling us that we're entering an era of unprecedented biological possibility, where the ancient dream of conquering disease, feeding the hungry, and healing the earth has found expression in the precise language of genetic code. But they're also reminding us that with great power comes great responsibility, and that the choices we make about how to use these remarkable tools will echo through generations, shaping not just the trajectory of human health and capability, but the very future of life on Earth. As we continue this gentle journey through the landscape of genetic possibility, we find ourselves not just observers of scientific progress, but participants in decisions that will define what it means to be human in an age where biology itself has become technology, and the code of life has become as malleable as words on a page, waiting to be rewritten with wisdom, compassion, and hope for a better tomorrow. The question that haunts many sleepless nights isn't whether we should proceed with CRISPR, but how we proceed with wisdom. As we navigate this delicate balance between scientific possibility and ethical responsibility, we find ourselves writing the rules as we go, crafting guidelines for technology that moves faster than our collective ability to fully comprehend its implications. International scientific organizations have begun establishing frameworks for CRISPR research, creating what some call a moral GPS for genetic exploration. These guidelines attempt to distinguish between acceptable and unacceptable applications, drawing lines in the genetic sand that researchers around the world can follow. But unlike traditional medical regulations that govern well understood treatments, CRISPR guidelines must anticipate applications that haven't been invented yet, preparing for possibilities we can barely imagine. The World Health Organization has created a registry for human genome editing research, essentially a global logbook where scientists must record their intentions before beginning studies involving genetic modifications to human embryos, eggs, or sperm. It's a transparency measure designed to prevent the kind of secretive research that shocked the world in 2018, ensuring that the international community can monitor and evaluate proposed studies before they begin. But governance becomes infinitely more complex when we consider the global nature of genetic research. A modification made in one laboratory could theoretically spread through natural reproduction to populations worldwide. A genetically edited organism released in one ecosystem could migrate across borders, carrying its artificial genes into territories that never consented to the experiment. These realities demand international cooperation on a scale rarely seen in scientific regulation. The challenge is particularly acute because different cultures and societies have vastly different comfort levels with genetic modification. What one nation considers therapeutic intervention, another might view as unacceptable tampering with human nature. These differences aren't merely academic disagreements, they're rooted in deep philosophical, religious, and cultural beliefs about the relationship between humans, nature, and the divine. Consider how different societies approach the concept of genetic enhancement. Some cultures emphasize individual choice and personal improvement, viewing genetic modifications as tools for self betterment, similar to education or exercise. Others prioritize community harmony and natural order, seeing genetic changes as potentially disruptive to social balance or spiritual well being. These perspectives shape not only what research is permitted, but how the results are interpreted and applied. Religious traditions add layers of complexity that scientists must navigate with particular sensitivity. Some faiths view genetic editing as a divine gift, a tool provided by providence for healing and improvement. Others see it as hubris, an inappropriate attempt to assume powers that belong only to higher authority. Still others focus on the intentions behind genetic modifications, blessing those aimed at healing while questioning those designed for enhancement or advantage. These diverse perspectives create a patchwork of regulations and attitudes that researchers must navigate carefully. A therapy that's celebrated in one country might be prohibited in another. A research approach that's considered ethical in one culture might be viewed as morally problematic in another. This regulatory diversity could lead to what ethicists call jurisdiction shopping, where researchers move their work to locations with more permissive oversight. The democratic process of making decisions about CRISPR becomes particularly challenging because the science is often too complex for general public understanding, yet the implications are too important to leave entirely to experts. How do you conduct meaningful public discourse about genetic modifications when most people lack the scientific background to fully evaluate the risks and benefits? How do you balance expert knowledge with democratic values when the stakes include the future of human evolution? Some countries have established citizen panels or consensus conferences where randomly selected members of the public spend weeks learning about genetic technologies and then provide recommendations to policymakers. These deliberative democracy approaches attempt to bridge the gap between expert knowledge and public values, creating forums where complex scientific issues can be discussed by informed citizens rather than just specialists. The economic dimensions of CRISPR governance add another layer of consideration. The development of genetic therapies requires enormous investments in research, testing, and regulatory approval. Companies that make these investments naturally expect to recoup their costs and earn profits, but genetic modifications raise unique questions about ownership and access. Can you patent a genetic modification? Should life saving genetic therapies be treated as public goods or private commodities? The patent landscape around CRISPR has become a battleground where universities, companies, and inventors fight over who owns the rights to various aspects of the technology. These legal battles aren't just about money, they're about who controls access to genetic therapies and how quickly new treatments can reach patients. A therapy that's tied up in patent disputes might be delayed for years, while lawyers argue over intellectual property rights. Access and equity concerns become particularly acute when we consider the global distribution of genetic diseases and economic resources. Many of the conditions that could benefit most from CRISPR therapies are prevalent in developing countries that lack the infrastructure or resources to develop or afford genetic treatments. This creates a moral paradox where the people who need genetic therapies most are least likely to receive them. Some researchers and organizations are working to address these equity concerns through open source approaches to genetic technology, sharing research tools and techniques freely rather than restricting them through patents. Others are developing tiered pricing systems where genetic therapies cost less in poorer countries, or creating research partnerships that ensure new treatments are accessible in the regions where they're most needed. The question of genetic enhancement versus therapy continues to generate intense debate among ethicists, policymakers, and the public. While most people agree that using CRISPR to treat serious genetic diseases is ethically acceptable, opinions diverge sharply when it comes to modifications that enhance normal human capabilities. Should parents be allowed to edit their children's genes to improve intelligence, athletic ability, or disease resistance beyond typical human ranges? These enhancement questions become even more complex when we consider their social implications. If genetic enhancements become available, will parents feel pressured to modify their children to keep up with enhanced peers? Will enhanced individuals, guess, have unfair advantages in education, employment, or other areas of life? Could genetic enhancement create new forms of discrimination or social stratification based on genetic modifications? The concept of genetic authenticity adds another philosophical dimension to these discussions. If we can edit our genes, what makes us genuinely human? Are we somehow less authentic if our capabilities come from genetic modifications rather than natural inheritance and personal effort? These questions don't have clear answers, but they influence how people think about the acceptability of genetic enhancements. Environmental governance of CRISPR applications presents unique challenges because genetic modifications released into ecosystems could have effects that spread far beyond their original location. Unlike medical applications that affect only consenting individuals, environmental releases could impact entire species, ecosystems, and even global environmental systems without the consent of affected communities. The development of gene drives, genetic modifications designed to spread rapidly through wild populations, has prompted calls for extremely cautious governance approaches. Some scientists have proposed moratoriums on environmental releases of gene drives until governance frameworks can be established. Others argue that the potential benefits of gene drives for controlling disease vectors or invasive species are too important to delay indefinitely. International environmental treaties provide some frameworks for governing genetic modifications that could cross borders, but these agreements were written before CRISPR existed and may not adequately address the unique challenges of genetic technologies. New international agreements or modifications to existing treaties may be needed to ensure that environmental applications of CRISPR are governed appropriately. The precautionary principle, which suggests that lack of full scientific certainty shouldn't postpone measures to prevent potential harm, plays a central role in discussions about environmental applications of CRISPR. However, applying this principle to genetic technologies is complicated because both action and inaction carry risks. Releasing genetically modified organisms could have unintended consequences, but failing to use genetic tools to address environmental crises could also lead to serious harm. Public engagement becomes particularly important for environmental applications because everyone shares the global environment that could be affected by genetic modifications. Unlike medical applications, where individuals can choose whether to undergo genetic treatments, environmental releases, affect everyone, whether they consent or not. This creates moral obligations for inclusive decision making processes that consider diverse perspectives and values. The governance challenges become even more complex when we consider future applications of CRISPR that we can barely imagine today. How do you regulate technologies that don't yet exist? How do you prepare for applications that current science fiction writers haven't even dreamed up? The history of technology suggests that the most transformative applications often emerge unexpectedly, making governance a continuous process of adaptation and learning. Adaptive governance approaches attempt to address this challenge by creating flexible regulatory frameworks that can evolve as technologies develop. Rather than trying to anticipate every possible application, these approaches focus on establishing principles and processes that can guide decision making as new possibilities emerge. They emphasize ongoing monitoring, evaluation, and adjustment rather than fixed rules that might become obsolete. The role of scientific advisory bodies becomes crucial in these adaptive governance systems. These groups of experts help policymakers understand new developments, assess risks and benefits, and recommend appropriate regulatory responses. However, the composition and independence of these advisory bodies can significantly influence their recommendations, making their selection and oversight important governance considerations. International cooperation remains perhaps the greatest challenge in governing CRISPR technologies. Genetic modifications don't respect national borders, and the global nature of scientific research means that governance approaches in one country can affect the entire world. Building international consensus on appropriate governance frameworks requires navigating different legal systems, cultural values, and political priorities. Some international organizations are working to facilitate these discussions by creating forums where different countries can share perspectives and work toward common approaches. The Organization for Economic Cooperation and Development, the World Health Organization, and various United Nations agencies have all engaged with CRISPR governance issues, though their authority to enforce agreements remains limited. The speed of scientific development continues to challenge governance systems that were designed for slower moving technologies. By the time regulatory agencies complete their assessments of one CRISPR application, researchers have often moved on to entirely new approaches that require fresh evaluation. This creates pressure to streamline regulatory processes without compromising safety or ethical oversight. Some experts have proposed regulatory sandboxes for genetic technologies, controlled environments where new applications can be tested with reduced regulatory barriers but enhanced monitoring and oversight. These approaches could allow beneficial applications to move forward more quickly while still protecting against potential harm. The challenge of governing CRISPR is ultimately about more than just regulating a particular technology. It's about how democratic societies make decisions about powerful technologies that could reshape fundamental aspects of human existence. It's about balancing innovation with precaution, individual freedom with collective responsibility, national sovereignty with global cooperation. As we continue to develop and apply CRISPR technologies, the governance frameworks we create will shape not only how these tools are used, but who benefits from them and what values guide their application. These decisions will echo through generations, influencing the trajectory of human health, the health of our planet, and the meaning of human flourishing in an age of genetic possibility. The whispers of science remind us that governance isn't an obstacle to scientific progress, but an essential companion to it, ensuring that our remarkable ability to edit the code of life serves the common good and reflects our deepest values about what it means to be human in a world where biology itself has become technology. The laboratory where CRISPR was first harnessed as a tool for genetic editing was quiet, filled with the gentle hum of equipment and the soft rustle of researchers moving carefully among their experiments. But the implications of their work were anything but quiet. They were crafting the first sentences in a new chapter of human capability, one that would ripple outward from their benches to touch lives across the globe in ways they were only beginning to understand. Today, that ripple has become a wave. Clinical trials are underway around the world, testing CRISPR therapies for conditions that have long been considered incurable. Patients with sickle cell disease, beta thalassemia, and certain cancers are among the first to experience the promise of genetic medicine. Their stories, still being written, represent the human face of scientific breakthrough. Take Victoria Gray, one of the first patients to receive CRISPR therapy for sickle cell disease. For years, her life was punctuated by painful crises, hospitalizations, and the constant awareness that her genetic inheritance carried a burden of suffering. The therapy involved extracting her bone marrow cells, editing them with CRISPR to produce healthy hemoglobin, and returning them to her body. Months later, her blood showed signs of transformation, producing the healthy red blood cells that had been absent since birth. Her story is remarkable, not just for its medical success, but for what it represents about the intersection of cutting edge science and deeply personal human experience. In her cells, CRISPR didn't just correct a genetic error, it rewrote a life story that had been shaped by genetic disease. The molecular scissors that cut and pasted DNA sequences also cut through decades of medical limitation and pasted in new possibilities for health and wholeness. But Victoria's story also highlights the current limitations of CRISPR medicine. The therapy required weeks of hospitalization, carried significant risks, and cost hundreds of thousands of dollars. While it may have cured her disease, it's not yet accessible to the millions of other people worldwide who suffer from sickle cell disease, particularly in sub-Saharan Africa, where the condition is most common, but resources for advanced medical treatments are most scarce. This disparity between promise and access runs through many current applications of CRISPR therapy. The first approved CRISPR treatments are marvels of precision medicine, but they're also expensive, complex procedures available primarily in wealthy countries with advanced medical infrastructure. The democratic promise of genetic healing remains largely unrealized, creating ethical tensions between what's possible and what's accessible. Research teams around the world are working to address these accessibility challenges through various approaches. Some are developing simpler delivery methods that could make CRISPR therapies easier to administer in less sophisticated medical settings. Others are exploring ways to reduce costs through more efficient manufacturing processes or open source approaches to genetic therapy development. The field of in vivo gene editing, where CRISPR modifications are made directly in the patient's body rather than in extracted cells, holds particular promise for increasing accessibility. Instead of requiring complex cell extraction and reinfusion procedures, in vivo approaches could potentially be delivered through simple injections, making genetic therapies available in many more healthcare settings. Early trials of in vivo CRISPR therapies are showing promising results for treating certain forms of inherited blindness, where CRISPR components can be injected directly into the eye to correct genetic defects in retinal cells. The elegance of this approach lies in its directness, editing genes in their natural cellular environment rather than requiring the complex cellular engineering of ex vivo approaches. But perhaps the most transformative potential of CRISPR lies not in treating diseases we already understand, but in revealing new possibilities for human health and capability. As our knowledge of genetics expands, researchers are discovering genes associated with resistance to HIV infection, protection from heart disease, enhanced cognitive function, and even extended lifespan. The possibility of editing these protective genetic variants into human genomes opens up entirely new categories of preventive medicine. Imagine visiting your doctor not to treat a disease, but to prevent one through genetic modification. Instead of taking daily medications to control cholesterol, you could receive a one time genetic edit that permanently reduces your cardiovascular risk. Instead of worrying about your family history of Alzheimer's disease, you could have protective genetic variants installed that significantly reduce your chances of developing dementia. These possibilities remain largely theoretical, but they're grounded in real genetic discoveries. Researchers have identified people with natural genetic mutations that protect them from HIV infection, heart disease, and other conditions. CRISPR could potentially allow us to share these protective mutations with others, democratizing genetic advantages that were previously limited to small numbers of naturally lucky individuals. The concept of genetic vaccination represents another frontier where CRISPR could revolutionize medicine. Instead of introducing weakened pathogens to stimulate immune responses, genetic vaccines could directly edit immune cells to recognize and respond to specific threats. This approach could potentially provide more durable immunity while avoiding the risks associated with traditional vaccine approaches. Cancer treatment continues to be one of the most active areas of CRISPR research, with approaches that go far beyond the CAR T cell therapies already in clinical use. Researchers are exploring ways to edit tumor suppressor genes directly in cancer cells, essentially reprogramming them to stop dividing and undergo programmed cell death. Others are working on editing immune cells to overcome the various tricks that tumors use to evade immune surveillance. Or the possibility of using CRISPR to address aging itself remains one of the most intriguing but speculative applications. As researchers identify genetic factors that contribute to cellular aging, the possibility emerges of editing these factors to slow or even reverse aspects of the aging process. While we're still far from a genetic fountain of youth, early research in laboratory animals suggests that targeted genetic modifications can extend healthy lifespan. Agricultural applications of CRISPR continue to expand as researchers explore ways to address the challenges of feeding a growing global population in a changing climate. Beyond the obvious applications of improving crop yields and pest resistance, scientists are developing crops with enhanced nutritional profiles, longer shelf lives, and reduced environmental impacts. The development of perennial grains represents one particularly ambitious application of CRISPR in agriculture. Most of our major food crops are annuals that must be replanted each year, requiring significant inputs of fuel, fertilizer, and labor. By editing the genes that control plant life cycles, researchers hope to create perennial versions of wheat, rice, and corn that would regrow each year from established root systems, dramatically reducing the environmental impact of agriculture. Nitrogen fixation, the process by which certain bacteria convert atmospheric nitrogen into forms that plants can use, represents another target for agricultural CRISPR applications. Currently, most crops require large amounts of nitrogen fertilizer, which contributes to water pollution and greenhouse gas emissions. Researchers are working to edit crop plants to either host nitrogen fixing bacteria or perform nitrogen fixation themselves, potentially reducing the environmental impact of agriculture while maintaining high yields. The intersection of CRISPR and climate change extends beyond agriculture to encompass broad environmental applications. Scientists are exploring ways to use genetic modifications to help ecosystems adapt to changing conditions, from coral reefs that can survive in more acidic oceans to forests that can withstand higher temperatures and altered precipitation patterns. Carbon capture and storage represents another potential environmental application of CRISPR technology. Researchers are working to edit plants and microorganisms to capture and store carbon more efficiently, potentially contributing to efforts to reduce atmospheric carbon dioxide levels. While these approaches are still largely experimental, they represent the kind of biological solutions that could complement technological approaches to climate change mitigation. The concept of assisted evolution using genetic modifications to help species adapt to environmental changes more quickly than natural selection would allow is gaining attention among conservation biologists. This approach could potentially help endangered species survive in changing environments or help key species adapt to new conditions that threaten ecosystem stability. However, these environmental applications also highlight the need for extremely careful consideration of ecological impacts. Unlike medical applications that affect only consenting individuals, environmental releases of genetically modified organisms could have effects that spread through ecosystems in unpredictable ways. The interconnectedness of natural systems means that modifying one species could have cascading effects throughout entire ecological communities. The development of containment strategies for genetically modified organisms remains an active area of research. Scientists are working on genetic modifications that would prevent modified organisms from surviving outside of controlled environments, ensuring that experimental organisms can't escape into wild populations. These molecular containment approaches could make environmental applications of CRISPR safer by providing multiple layers of biological containment. Gene drives, genetic modifications designed to spread rapidly through wild populations, represent both the greatest promise and the greatest concern in environmental applications of CRISPR. While gene drives could potentially eliminate disease-carrying mosquitoes or control invasive species, They could also have irreversible effects on entire ecosystems if they spread beyond their intended targets. Current research on gene drives focuses heavily on developing control mechanisms that could halt or reverse their spread if unintended consequences emerge. These include temporal controls that cause gene drives to weaken over time, spatial controls that limit their geographic spread, and molecular controls that can be activated to counteract their effects. The global nature of many environmental challenges means that environmental applications of CRISPR will likely require unprecedented levels of international cooperation. Climate change, biodiversity loss, and ecosystem degradation cross national boundaries, and genetic approaches to addressing these challenges will need to be coordinated across countries and regions. International research collaborations are already emerging to address these challenges, bringing together scientists from different countries to work on shared environmental problems. These collaborations not only pool scientific expertise, but also help ensure that environmental applications of CRISPR consider diverse perspectives and values from different cultures and societies. The timeline for many environmental applications remains uncertain as researchers balance the potential benefits of genetic interventions against the need for extremely careful safety testing. Unlike medical applications, where the benefits to individual patients can justify accepting certain risks, environmental applications affect entire ecosystems and future generations, requiring higher standards of evidence and broader consensus before implementation. Public engagement around environmental applications of CRISPR has become increasingly important as the technology moves from laboratory research toward potential real world applications. Environmental organizations, indigenous communities, and other stakeholders are demanding meaningful participation in decisions about genetic modifications that could affect shared natural resources. The challenges of governing environmental applications of CRISPR highlight the need for new models of decision making that can accommodate scientific uncertainty, diverse stakeholder perspectives, and the global nature of environmental systems. Traditional regulatory approaches that focus on assessing individual products or applications may be inadequate for genetic technologies that could have system wide effects. As we look toward the future of CRISPR applications, we see a technology that continues to evolve and surprise us with new possibilities. The bacterial immune system that inspired CRISPR has given us tools that reach far beyond what its microbial inventors ever needed, allowing us to edit not just viral DNA, but the genetic foundation of life itself. The whispers of science tell us that we're still in the early chapters of the CRISPR story, with most of its transformative applications yet to be written. Each new discovery, each successful treatment, each solved challenge opens doors to possibilities we haven't yet imagined, reminding us that the greatest revolutions often begin with the quietest observations in the most unexpected places. The story of CRISPR is also a story about the beautiful unpredictability of scientific discovery, where a seemingly mundane observation about bacterial behavior led to one of the most powerful tools in human history. It reminds us that we never know where the next breakthrough might come from, or how a curious mind investigating something completely unrelated might stumble upon the key to solving humanity's greatest challenges. In laboratories around the world today, researchers are making observations that might seem trivial or esoteric, but could contain the seeds of tomorrow's miracles. Someone studying the way certain bacteria communicate with each other might discover new approaches to treating neurological diseases. A researcher investigating how plants respond to stress might unlock new strategies for human longevity. A scientist examining the genetics of extremophile organisms living in harsh environments might find the key to helping humans adapt to the challenges of space travel or climate change. This interconnectedness of biological systems means that advances in CRISPR often come from unexpected directions. The development of base editing, which allows scientists to change single letters of DNA without cutting the double helix, emerged from studies of how certain enzymes naturally modify genetic sequences. Prime editing, which provides even greater precision in genetic modifications, was inspired by understanding how cells naturally repair DNA damage. These technical advances continue to expand what's possible with genetic editing. Scientists are now developing CRISPR systems that can edit RNA instead of DNA, providing temporary modifications that don't permanently alter the genetic code. Others are working on epigenetic editors that can turn genes on or off without changing their underlying sequences, like adjusting the volume on different genetic programs without rewriting the music itself. The miniaturization of CRISPR systems represents another frontier that could dramatically expand their applications. Researchers are engineering smaller, more efficient versions of the molecular scissors that could be delivered more easily to hard to reach tissues or used in resource limited settings. These compact genetic editors could make CRISPR therapies more accessible and affordable around the world. Multiplexed editing, the ability to make multiple genetic changes simultaneously, is opening up possibilities for addressing complex genetic conditions that involve multiple genes. Instead of editing one gene at a time, scientists can now orchestrate comprehensive genetic modifications that address entire pathways or systems. It's like conducting a molecular symphony where multiple instruments play in harmony to create therapeutic effects that no single modification could achieve. The development of inducible CRISPR systems adds another layer of control to genetic modifications. These systems remain dormant until activated by specific signals, allowing researchers to control when and where genetic changes occur. A therapy could be administered and then activated only when needed, or genetic modifications could be turned on or off in response to changing conditions in the body. Tissue specific editing represents yet another refinement that could make CRISPR therapies safer and more effective. By engineering CRISPR systems that only functions in certain types of cells, scientists can ensure that genetic modifications occur only where they're intended, reducing the risk of off-target effects in healthy tissues. The convergence of CRISPR with other emerging technologies continues to create new possibilities that neither approach could achieve alone. Combining genetic editing with regenerative medicine could allow us to create replacement tissues and organs that are perfectly matched to individual patients, eliminating the problems of immune rejection that plague current transplant approaches. The integration of CRISPR with advanced imaging technologies is enabling real-time monitoring of genetic modifications as they occur. Scientists can now watch genetic changes unfold in living cells and organisms, providing immediate feedback about the effectiveness and safety of different editing approaches. This visual confirmation adds a new dimension to genetic research, allowing researchers to see their molecular interventions in action. Artificial intelligence continues to play an increasingly important role in CRISPR research, helping scientists design more effective guide RNAs, predict potential side effects, and identify new therapeutic targets. Machine learning algorithms can analyze vast databases of genetic information to find patterns that human researchers might miss, suggesting new approaches to genetic therapy that no one had previously considered. The democratization of CRISPR technology through simplified delivery systems and reduced costs is beginning to make genetic editing accessible in unexpected places. University laboratories in developing countries are using CRISPR to address local health challenges, from developing crops adapted to regional growing conditions to investigating genetic variants that affect disease susceptibility in different populations. Community laboratories and citizen science initiatives are also beginning to explore CRISPR applications, though with appropriate safety measures and oversight. These grassroots efforts could lead to innovations that might not emerge from traditional research institutions, bringing diverse perspectives and creativity to genetic research. The educational implications of widespread CRISPR adoption are profound. As genetic editing becomes more common in medicine and agriculture, public understanding of genetics and biotechnology becomes increasingly important. People need to understand enough about these technologies to make informed decisions about their own care and to participate meaningfully in democratic discussions about how genetic tools should be regulated and applied. Nutzt. Medical schools and healthcare training programs are already adapting their curricula to include genetic editing technologies. Tomorrow's doctors will need to understand not just how to diagnose and treat diseases, but how to counsel patients about genetic therapies and navigate the ethical complexities of genetic enhancement. Agricultural extension programs are similarly adapting to help farmers understand and utilize genetically edited crops. As these technologies become more accessible, farmers around the world will need training and support to make informed decisions about which genetic modifications might benefit their specific growing conditions and market needs. The economic transformation driven by CRISPR extends far beyond the biotechnology industry itself. Traditional pharmaceutical companies are partnering with genetic editing startups, creating new business models that focus on one-time curative therapies rather than chronic disease management. Insurance companies are grappling with how to cover genetic therapies that might prevent diseases decades before they would have occurred. Agricultural markets are being reshaped by crops with enhanced nutritional profiles, longer shelf lives, and reduced environmental impacts. Food companies are exploring how genetically edited ingredients might allow them to create healthier, more sustainable products. Environmental service companies are emerging to provide genetic solutions to ecological challenges. The job market is evolving to include entirely new categories of employment related to genetic technologies. Genetic counselors, bioethicists, regulatory specialists, and genetic technicians represent just a few of the professional roles that are expanding as CRISPR applications multiply. The intersection of biology, technology, and data science is creating career paths that didn't exist a generation ago. International trade relationships are also being influenced by different national approaches to genetic modification. Countries with more permissive regulations for genetically edited crops might gain competitive advantages in agricultural exports, while those with stricter oversight might find themselves importing genetic innovations developed elsewhere. The geopolitical implications of CRISPR leadership are becoming increasingly apparent as nations recognize the strategic importance of genetic technologies. Countries are investing heavily in genetic research infrastructure, not just for economic benefits, but for national security considerations. The ability to quickly develop genetic countermeasures to biological threats or to ensure food security through genetically adapted crops could become crucial elements of national resilience. Research collaborations and competition between nations in genetic technologies mirror broader patterns of international cooperation and rivalry. While scientific knowledge benefits from open collaboration, the applications of that knowledge can create competitive advantages that nations are reluctant to share freely. The global nature of genetic challenges, from pandemic diseases to climate change impacts on agriculture, creates natural incentives for international cooperation in CRISPR research. However, differences in regulatory approaches, ethical frameworks, and economic interests can complicate these collaborative efforts. As we contemplate the future trajectory of CRISPR technology, we find ourselves imagining possibilities that stretch the boundaries of current understanding. The tools that began as bacterial immune systems might eventually allow us to engineer biological solutions to challenges we can barely envision today. Climate adaptation, space exploration, longevity extension, cognitive enhancement, ecosystem restoration, and countless other applications await discovery by researchers who are just beginning their careers. The children who are growing up today will inherit a world where genetic editing is commonplace, where many of the diseases that have plagued humanity for millennia have been eliminated, and where the boundary between natural and artificial biology has become beautifully blurred. They will face questions about genetic identity, enhancement, and responsibility that we can only begin to anticipate. Yet perhaps the most remarkable aspect of the CRISPR revolution is not its technical sophistication, but its reminder of the fundamental unity of life. The same genetic code that we can now edit with precision is shared by every living thing on Earth, from the bacteria that inspired CRISPR to the humans who now wield it as a tool for healing and improvement. This universality suggests that our growing power to edit life comes with a corresponding responsibility to use that power wisely, not just for our own benefit, but for the health and flourishing of the entire community of life on Earth. The molecular scissors that can cut and paste genetic sequences can also help us cut through old assumptions about what's possible and paste together new visions of human potential and planetary health. The whispers of science remind us that we stand at a threshold where ancient bacterial wisdom meets human ingenuity, where the mechanisms that have protected microscopic life for billions of years have been transformed into tools for healing, feeding, and nurturing life on a global scale, carrying us gently into a future where the code of life itself has become our partner in writing the next chapter of existence on Earth. As we drift toward the conclusion of our gentle exploration through the molecular landscapes of genetic possibility, we find ourselves returning to perhaps the most profound question of all. What does it mean to hold the power to rewrite life itself? In laboratories bathed in soft fluorescent light, researchers continue their quiet work, each experiment adding another brushstroke to a portrait of human capability that grows more remarkable with each passing day. The story of CRISPR that we've traced together tonight, from ancient bacterial battlefields to cutting edge clinical trials, reflects something deeper about the nature of scientific discovery and human curiosity. It reminds us that the most transformative breakthroughs often emerge not from grand design, but from patient observation, from scientists curious enough to ask why bacteria behave in seemingly peculiar ways, dedicated enough to pursue answers even when the practical applications weren't immediately obvious. Jennifer Doudna, one of CRISPR's pioneering architects, has often spoken about the moment she realized the broader implications of their bacterial immune system research. It wasn't a dramatic eureka moment, but rather a gradual dawning, like watching sunrise slowly illuminate a landscape that had been hidden in darkness. The recognition that this ancient microbial defense mechanism could be repurposed as a universal genetic editing tool came through careful experimentation and collaborative thinking, scientists building upon each other's insights in the patient, methodical way that characterizes the best of human inquiry. This collaborative nature of CRISPR's development offers a beautiful counterpoint to popular images of solitary genius working in isolation. The technology emerged from a global community of researchers, each contributing pieces to a puzzle that none could have solved alone. Microbiologists studying bacterial behavior, biochemists investigating protein structures, molecular biologists exploring gene regulation, and countless others all played roles in the discovery and refinement of genetic editing tools that now hold such promise for human welfare. The international character of this research collaboration points towards something hopeful about human nature itself. Despite political tensions and economic competition between nations, scientists continue to share knowledge, build upon each other's work, and collaborate across cultural and linguistic boundaries in pursuit of understanding that benefits all humanity. The bacterial immune systems that inspired CRISPR don't recognize national borders, and neither does the scientific curiosity that transformed them into tools for healing. Yet, as we've explored throughout our journey tonight, this remarkable power comes wrapped in equally remarkable responsibility. The ability to edit genes carries with it obligations that extend far beyond individual patients or research laboratories. Every genetic modification, whether in a human cell, a crop plant, or a wild organism, sends ripples through interconnected systems of biology, society, and ethics that we're still learning to navigate. The pace of CRISPR development sometimes outstrips our collective wisdom about how to use these tools appropriately. Like explorers mapping uncharted territory, we often find ourselves making decisions with incomplete information, guided by principles and values that must evolve as our capabilities expand. The ethical frameworks we're developing for genetic editing represent humanity's attempt to ensure that our growing power serves our deepest values rather than undermining them. Perhaps one of the most striking aspects of the CRISPR revolution is how it has democratized genetic research making sophisticated gene editing accessible to laboratories around the world. This democratization brings both opportunities and challenges. On one hand, it means that innovative applications can emerge from unexpected places, that researchers in developing countries can contribute to genetic solutions for local problems, and that the benefits of genetic technology might eventually reach communities that have been excluded from previous medical advances. On the other hand, the widespread availability of genetic editing tools raises concerns about oversight and safety. When powerful technologies become accessible to many users, ensuring that they're used responsibly becomes more complex. The scientific community has responded by developing guidelines and training programs, but the challenge of maintaining safety and ethical standards across thousands of research groups worldwide remains formidable. The educational implications of living in a CRISPR enabled world extend beyond training scientists and medical professionals. As genetic editing becomes more common in medicine and agriculture, citizens need sufficient understanding of these technologies to make informed decisions about their own lives and to participate meaningfully in democratic discussions about regulation and policy. This need for genetic literacy represents a new dimension of civic education, preparing people to engage thoughtfully with technologies that could reshape fundamental aspects of human existence. The stories of patients who have already benefited from CRISPR therapies offer glimpses of the human dimension of genetic medicine. Beyond the technical details of guide RNAs and molecular scissors lie individual narratives of suffering transformed into hope, of genetic destinies rewritten through scientific ingenuity and medical courage. These personal stories remind us that the ultimate purpose of genetic research isn't abstract knowledge, but human flourishing, the relief of suffering, and the expansion of possibility for people whose lives have been constrained by genetic limitations. Yet, even as we celebrate these early successes, we must acknowledge the challenges that remain. Current CRISPR therapies are expensive, complex, and available primarily in wealthy countries with advanced medical infrastructure. The gap between what's technically possible and what's practically accessible represents one of the most pressing challenges facing genetic medicine. Researchers and policymakers are working to address these disparities, but ensuring equitable access to genetic therapies will require sustained effort and creative thinking about healthcare delivery and financing. The agricultural applications of CRISPR offer different but equally significant opportunities to improve human welfare. As climate change creates new challenges for food production, genetically edited crops could help maintain food security while reducing the environmental impact of agriculture. The development of crops that can thrive in changing conditions, require fewer inputs, or provide enhanced nutrition could benefit billions of people, particularly in developing countries where food security remains precarious. However, the deployment of genetically edited crops also raises questions about agricultural sustainability, farmer autonomy, and corporate control of food systems. The history of agricultural technology adoption suggests that new innovations don't automatically benefit all participants in food systems equally. Ensuring that CRISPR applications in agriculture serve small farmers and vulnerable populations, as well as large agribusiness, will require careful attention to policy design and implementation. The environmental applications of CRISPR perhaps carry the highest stakes and the greatest uncertainties. The possibility of using genetic modifications to address climate change, restore ecosystems, or conserve endangered species offers hope for tackling some of humanity's most pressing challenges. However, the interconnectedness of natural systems means that genetic interventions could have far reaching and unpredictable consequences. The decision to release genetically modified organisms into the environment represents a form of planetary experimentation that demands extraordinary caution and wisdom. The concept of reversibility becomes particularly important when considering environmental applications. Unlike medical treatments that affect only consenting individuals, environmental releases of genetically modified organisms could create changes that are difficult or impossible to undo. This irreversibility places a heavy burden of responsibility on current generations to make decisions that will affect the planet's biological heritage for centuries to come. As we look toward the future of CRISPR technology, we see a field that continues to surprise us with new capabilities and applications. The bacterial immune system that inspired the original discovery has given rise to an entire toolkit of genetic editing approaches, each refined for specific applications and challenges. Base editors that can change single letters of DNA without cutting the double helix, prime editors that can insert or delete specific sequences with unprecedented precision, and epigenetic editors that can modify gene expression without altering underlying DNA sequences, all represent evolutionary steps beyond the original CRISPR-Cas9 system. These technical advances are making genetic editing more precise, more versatile, and potentially safer. As the tools become more sophisticated, they open up new possibilities for treating genetic conditions that were previously considered beyond reach. Complex genetic diseases involving multiple genes might become treatable through coordinated editing approaches. Subtle genetic modifications that enhance human health or capability without crossing into problematic enhancement territory might become feasible and socially acceptable. The convergence of CRISPR with other emerging technologies, artificial intelligence, nanotechnology, synthetic biology, advanced imaging creates possibilities that no single approach could achieve alone. These technological synergies suggest that the most transformative applications of genetic editing may still lie ahead, emerging from combinations of tools and techniques that we're only beginning to explore. Artificial intelligence, in particular, is becoming an increasingly important partner in genetic research. Machine learning algorithms can help design more effective guide RNAs, predict potential side effects, and identify new therapeutic targets in ways that would be impossible for human researchers alone. As AI systems become more sophisticated and genetic databases grow larger, the partnership between human creativity and artificial intelligence could accelerate the pace of genetic discovery dramatically. The global nature of genetic challenges creates natural incentives for international cooperation in CRISPR research and application. Pandemic diseases, climate change impacts on agriculture, and biodiversity loss all cross national boundaries, requiring coordinated responses that transcend political divisions. The scientific community's tradition of open collaboration provides a foundation for these international efforts, though differences in regulatory approaches and ethical frameworks can complicate cooperation. The development of global governance frameworks for genetic technologies remains an ongoing challenge. Unlike previous technologies that could be regulated primarily at national levels, genetic modifications can have effects that spread across borders and affect shared global resources, creating international agreements and institutions capable of overseeing genetic technologies while respecting national sovereignty and cultural differences requires diplomatic skill and scientific wisdom in equal measure. As we contemplate the long term trajectory of CRISPR development, we find ourselves imagining a future where genetic editing becomes as routine and accepted as other medical interventions that once seemed miraculous. The children growing up today may inhabit a world where genetic diseases are largely eliminated, where crops are precisely adapted to local growing conditions, and where the boundary between natural and designed biology has become so blurred as to be meaningless. Yet, even as we envision these technological possibilities, we must remember that the most important questions about CRISPR aren't technical but human. How do we ensure that genetic technologies serve human dignity rather than undermining it? How do we balance individual choice with collective responsibility? How do we preserve what's valuable about human nature while embracing the possibilities for improvement that genetic editing offers? These questions don't have simple answers, but they're questions that each generation must engage with as genetic technologies continue to evolve. The choices we make today about how to develop and deploy CRISPR will shape the options available to future generations, creating a legacy of genetic possibility or constraint that will endure long after current researchers have finished their work. The democratization of genetic technology through improved accessibility and reduced costs offers hope that the benefits of CRISPR won't be limited to wealthy individuals and nations. As delivery methods become simpler and manufacturing costs decrease, genetic therapies could potentially reach communities that have been excluded from previous medical advances. However, realizing this potential will require deliberate effort to ensure that genetic technologies are developed and distributed equitably. The role of public engagement in shaping the future of CRISPR cannot be overstated. While technical expertise is essential for developing genetic technologies safely and effectively, the decisions about how these tools should be used ultimately belong to the broader human community. Democratic societies need mechanisms for meaningful public participation in discussions about genetic editing, ensuring that diverse voices and values are heard in decisions that could affect everyone. Educational initiatives that help people understand genetic technologies without requiring advanced scientific training become increasingly important as CRISPR applications multiply. Citizens need sufficient knowledge to evaluate genetic therapies for themselves and their families, to participate in policy discussions about genetic research regulation, and to consider the broader implications of living in a genetically editable world. The economic transformations driven by CRISPR adoption will likely create both opportunities and disruptions across multiple sectors. Traditional approaches to medicine, agriculture, and environmental management may need to adapt to a world where genetic solutions become increasingly available and cost effective. These transitions could create new forms of employment while making others obsolete, requiring thoughtful planning to ensure that the benefits of genetic technology are broadly shared. The integration of genetic editing into existing healthcare systems presents both technical and social challenges. Medical professionals need training in genetic technologies. Healthcare institutions need infrastructure to support genetic therapies, and insurance systems need frameworks for covering genetic treatments that might prevent diseases decades before they would have occurred. These adaptations require coordination across multiple sectors and stakeholders. As we near the end of our gentle journey through the landscape of genetic possibility, we find ourselves returning to the fundamental wonder that drives scientific inquiry. The bacterial immune systems that inspired CRISPR represent just one example of the remarkable solutions that evolution has crafted over billions of years. Every living organism carries within its genes the accumulated wisdom of countless generations of adaptation and survival, and genetic editing technologies allow us to learn from this biological heritage in unprecedented ways. The universality of the genetic code means that insights gained from studying any organism can potentially inform our understanding of all life. The mechanisms that bacteria use to defend against viruses can be repurposed to treat human diseases. The strategies that plants use to survive drought can be transferred to crops growing in arid regions. The longevity genes found in some animals might eventually help humans live healthier, longer lives. This interconnectedness of all life suggests that our growing power to edit genes comes with responsibilities that extend beyond our own species. As we reshape the genetic landscape through CRISPR applications, we're also reshaping the evolutionary context within which all life exists. The modifications we make today could influence the trajectory of life on Earth for millions of years to come, making us temporary custodians of an evolutionary heritage that stretches back to the earliest forms of life. The ethical frameworks we develop for genetic editing must therefore consider not only immediate human welfare, but also our obligations to future generations and to the broader community of life on Earth. These considerations don't necessarily prohibit genetic modifications, but they do require us to proceed with humility, wisdom, and deep respect for the complexity and beauty of the living systems we're learning to modify. The story of CRISPR also reminds us of the importance of curiosity-driven research that pursues knowledge without immediate practical applications in mind. The scientists who first studied bacterial immune systems weren't trying to develop genetic therapies. They were simply curious about how bacteria defend themselves against viral infections. Yet their basic research laid the foundation for one of the most powerful biotechnologies ever developed. This connection between fundamental research and practical application suggests that supporting scientific curiosity broadly, even when the applications aren't immediately obvious, remains essential for continued progress in genetic technologies. The next breakthrough in genetic editing might come from studying extremophile organisms living in harsh environments, from investigating the genetics of aging in simple animals, or from exploring the molecular mechanisms of plant communication. We never know where scientific curiosity might lead us next. The collaborative nature of CRISPR development also offers lessons about how complex scientific challenges are best addressed. No single researcher or institution could have developed genetic editing technologies alone. Progress required collaboration across disciplines, institutions, and nations, with each contributor building upon the work of others in an ongoing conversation that continues today. This collaborative model becomes even more important as genetic technologies tackle increasingly complex challenges. Addressing genetic diseases that involve multiple genes, developing crops that can adapt to climate change, or using genetic modifications to restore damaged ecosystems all require expertise from multiple fields working together toward common goals. As we settle into the final moments of our exploration, we might reflect on what the CRISPR revolution tells us about human nature itself. Our species has always sought to understand and modify our environment to better meet our needs, from the development of agriculture to the invention of medicine. Genetic editing represents the latest chapter in this ongoing story of human ingenuity and adaptation. Yet CRISPR also represents something qualitatively different from previous technologies. For the first time in human history, we have the ability to consciously direct our own biological evolution, to make deliberate changes to the genetic heritage we pass on to future generations. This power brings us face to face with fundamental questions about human identity, enhancement, and responsibility that we're still learning to answer. The choices we make about how to use genetic editing technologies will reflect our deepest values about what it means to be human, what kinds of lives are worth living, and what obligations we have to each other and to the natural world. These are not technical questions, but moral and philosophical ones that require the wisdom of entire communities rather than just individual experts. Perhaps the most remarkable aspect of the CRISPR story is how it demonstrates the beautiful unpredictability of scientific discovery. A seemingly obscure observation about bacterial behavior led to tools that could transform medicine, agriculture, and environmental management in ways that the original researchers never imagined. This unpredictability suggests that maintaining scientific curiosity and supporting diverse research approaches remains essential even when we can't predict where they might lead. The whispers of science that have guided our journey tonight remind us that we live in an extraordinary moment in human history when ancient biological mechanisms discovered through patient scientific inquiry have been transformed into tools for healing, feeding, and nurturing life on a global scale. The bacterial immune systems that evolved millions of years ago to protect microscopic organisms have become instruments for protecting and enhancing human welfare, bridging the vast scales of biological organization from molecules to ecosystems. As you drift toward sleep tonight, perhaps you'll carry with you a sense of wonder at the remarkable journey we've taken together, from the molecular battlefields where bacteria and viruses wage their ancient wars, to the research laboratories where human ingenuity transforms biological wisdom into tools for healing. The same genetic code that CRISPR can now edit with such precision flows through every living thing on Earth, connecting us to the vast community of life that shares our planet and reminding us that our growing power to reshape biology comes with the profound responsibility to use that power wisely, gently, and with deep respect for the intricate beauty of the living world we're privileged to call home. The revolution in genetic editing that began with bacterial immune systems continues to unfold around us, writing new chapters in the story of human capability and responsibility, whispering promises of healing and hope while reminding us that the greatest scientific discoveries often emerge from the quietest observations in the most unexpected places, guided by nothing more than human curiosity and the patient desire to understand how life works at its most fundamental level. Sleep well, knowing that somewhere in laboratories around the world, researchers continue their gentle work with molecular scissors and genetic code, crafting therapies that might one day eliminate suffering, crops that could feed a hungry world, and solutions to challenges we haven't yet learned to imagine, all built upon the ancient wisdom of bacteria that learned long ago how to recognize friend from foe and defend the precious genetic information that makes life possible. The whispers of science will continue long after we've finished listening, carrying forward the conversation between human curiosity and natural wisdom that has brought us to this remarkable moment where the code of life itself has become our partner in writing the next chapter of existence on Earth, one gene at a time, one discovery at a time, one quiet breakthrough at a time, in the endless dance between what is and what could be, between the ancient and the innovative, between the microscopic and the magnificent. Sweet dreams, fellow traveler, in the gentle landscape of genetic wonder. May your rest be filled with the quiet satisfaction that comes from understanding something profound about the world we share. And may you wake tomorrow with a renewed appreciation for the remarkable fact that the same four-letter alphabet that spells out your existence also writes the stories of every bacterium, every blade of grass, and every creature that has ever drawn breath on this beautiful, interconnected planet we call home. The quiet revolution continues to unfold in ways both subtle and profound, each day bringing new whispers of possibility from laboratories where the impossible slowly becomes inevitable. As we reach the gentle conclusion of our molecular journey tonight, perhaps it's worth pausing to consider not just what CRISPR can do, but what it teaches us about the nature of discovery itself. There's something beautifully humbling about the fact that one of humanity's most powerful tools emerged from studying creatures so small we need microscopes to see them. Bacteria, those ancient survivors that have weathered every extinction event our planet has endured, developed their genetic defense systems long before complex life even existed. They were editing genes when the Earth was still a very different place, when the atmosphere held different gases and the continents sat in unfamiliar arrangements. In a sense, we haven't invented gene editing at all. We've simply learned to speak a language that life has been using for billions of years, a molecular conversation that predates human civilization by an almost incomprehensible span of time. Every CRISPR edit we make today uses tools that bacteria perfected when our planet was young, when the only witnesses to their molecular innovations were other microbes floating in ancient seas. This perspective offers a kind of comfort, doesn't it? The power we wield through CRISPR isn't foreign to life itself. It's life's own invention, borrowed and refined but fundamentally biological. We're not imposing artificial solutions on natural systems. We're participating in processes that have been shaping living things since the very beginning. The molecular scissors we use to heal genetic diseases are the same ones that helped bacteria survive in a world full of viral threats, generation after generation, eon after eon. But perhaps what's most remarkable about CRISPR isn't its technical elegance or its therapeutic promise, but the way it connects us to the deepest rhythms of existence. Every time a researcher designs a guide RNA or programs a Cas protein, they're engaging in the same fundamental activity that has driven evolution for billions of years: the careful, deliberate modification of genetic information to better suit changing circumstances. The difference, of course, is consciousness. Where evolution works through blind trial and error across vast spans of time, we can now work with intention, with foresight, with compassion for suffering, and hope for improvement. We can direct genetic changes towards specific goals, whether that's curing a child's genetic disease, helping crops survive in a changing climate, or preserving endangered species that might otherwise vanish forever. This consciousness brings with it both tremendous opportunity and tremendous responsibility. Every genetic modification we make is a choice, a decision about what kind of future we want to create. Unlike the random mutations that drive natural evolution, our CRISPR edits carry intention, values, hopes, and sometimes fears about what tomorrow might bring. The children being born today will grow up in a world where genetic editing is as commonplace as vaccination or surgery, where the question isn't whether we can modify genes, but whether we should, and how, and for whom. They'll inherit both the benefits of our early experiments with genetic healing and the consequences of our choices about how to use these powerful tools. Some of those children may themselves become the scientists who push CRISPR technology into territories we can barely imagine today. They might develop ways to edit genes with even greater precision, to address genetic challenges we haven't yet encountered, or to apply biological solutions to problems that don't even exist yet. The tools they create will likely make today's CRISPR systems seem quaint, like comparing a modern smartphone to the first telegraph. But whatever technical advances await us, the fundamental questions that CRISPR raises will likely persist. How do we balance individual benefit with collective risk? How do we ensure that genetic technologies serve justice rather than inequality? How do we preserve what's valuable about natural diversity while embracing the possibilities for improvement that genetic editing offers? These questions don't have final answers because they're not really technical questions at all. They're human questions reflecting our deepest values about life, dignity, suffering, and hope. Every generation will need to engage with them anew as our capabilities expand and our understanding deepens. The whispers of science that have guided our exploration tonight remind us that we're living through one of those rare moments in history when humanity's relationship with life itself is being fundamentally transformed. Like the development of agriculture, the invention of medicine, or the discovery of antibiotics, CRISPR represents a threshold crossing, a moment when what seemed impossible suddenly becomes not just possible, but inevitable. Yet, for all its revolutionary potential, CRISPR also connects us to something timeless and universal. The genetic code it edits is the same code that's been carrying life's instructions for billions of years. The cellular machinery it works with is the same machinery that built every living thing that has ever existed. The DNA it modifies carries within it the accumulated wisdom of countless generations of adaptation and survival. In editing genes, we're not stepping outside of nature, but deeper into it, not abandoning biological wisdom, but learning to read and write in its ancient language. We're becoming conscious participants in the ongoing conversation between life and its environment, between what is and what could be, between the challenges each generation faces and the solutions they develop to meet them. As our gentle journey draws to its close, perhaps the most important thing to remember is that CRISPR, for all its technical sophistication, is ultimately a tool in service of very simple human impulses: the desire to heal, to protect, to nurture, to improve. The same impulses that led our ancestors to develop medicines from plants, to select better seeds for planting, to care for the sick and vulnerable members of their communities. What's changed isn't our motivations, but our capabilities. We can now address genetic problems that previous generations could only observe and endure. We can prevent diseases before they manifest, enhance crops before they fail, and restore species before they vanish. The molecular precision of CRISPR gives us unprecedented power to act on our oldest and most fundamental impulses to make life better, safer, more abundant. But with that power comes the need for wisdom that matches our technical capabilities. The ability to edit genes must be coupled with deep thinking about when and how to use that ability. The precision of CRISPR must be matched by precision in our ethical reasoning, our policy making, our consideration of consequences that might unfold over decades or centuries. The bacterial immune systems that inspired CRISPR evolved over millions of years through countless cycles of attack and defense, threat and response. The bacteria that survived were those whose genetic defenses proved most effective, most adaptable, most resilient. In borrowing their tools, we've also inherited something of their patience, their persistence, their capacity to adapt and endure. Perhaps that's the deepest lesson CRISPR offers us, that the most powerful solutions often emerge not from dramatic breakthroughs, but from patient observation, careful experimentation, and the gradual accumulation of understanding. The researchers who first noticed strange repetitive sequences in bacterial genomes couldn't have imagined that their curiosity would lead to one of the most transformative technologies in human history. This reminds us to value curiosity for its own sake, to support research even when its applications aren't immediately obvious, to trust that patient inquiry into how the world works will eventually yield insights that benefit everyone. The next breakthrough might come from studying how plants communicate with soil microbes, how certain animals resist aging, or how extremophile organisms survive in impossible conditions. The interconnectedness of biological knowledge means that every discovery, no matter how seemingly abstract or esoteric, potentially contributes to our growing ability to understand and improve life. The boundary between basic science and applied technology becomes increasingly blurred as we realize that everything in biology is connected to everything else. As you settle into sleep tonight, dear listener, you might take comfort in knowing that around the world, researchers are continuing the patient work of discovery that brought us CRISPR. In laboratories illuminated by soft light, scientists are making observations, running experiments, asking questions that might seem simple but could unlock profound new understanding. Someone tonight is studying how bacteria communicate with each other, research that might lead to new approaches for treating infectious diseases. Someone else is investigating how certain plants survive in extreme cold, work that could help develop crops for changing climates. A researcher somewhere is examining the genetics of longevity in tiny worms, studies that might eventually help humans live healthier, longer lives. The great conversation between human curiosity and natural wisdom continues, carried forward by countless individuals united by wonder at the complexity and beauty of living systems. Each small discovery adds to our collective understanding. Each failed experiment teaches us something valuable. Each successful application opens doors to new possibilities we hadn't imagined. The genetic code that CRISPR allows us to edit with such precision is the same code that connects every living thing on Earth in an unbroken chain of inheritance, stretching back to the very origins of life. In learning to read and write in this ancient language, we're not just developing new medical treatments or agricultural innovations, we're participating in the ongoing story of life itself. Tonight, as you drift into dreams, the same molecular processes that inspired CRISPR are continuing in every cell of your body. DNA is being repaired, genes are being expressed, proteins are being made, all according to instructions written in the four-letter alphabet that every living thing shares. You are, in the most literal sense, a walking, breathing example of the genetic principles that make CRISPR possible. The whispers of science grow softer now as our exploration reaches its end, but they never truly stop. They continue in every laboratory where researchers peer into microscopes, in every greenhouse where edited plants grow, in every clinic where genetic therapies offer hope to patients who once had none. They echo in the questions asked by curious children, in the wonder felt by students encountering genetics for the first time, in the satisfaction of scientists whose work contributes to humanity's growing understanding of life. Sleep peacefully, knowing that you've touched, however briefly, one of the great stories of our time, how human ingenuity learned to speak the language of life itself, how bacterial wisdom became a tool for healing, how the humblest microorganisms taught us to rewrite the very code of existence. The revolution in genetic editing continues to unfold around us, gentle but unstoppable, promising and challenging, ancient in its origins, but futuristic in its applications. Tomorrow will bring new discoveries, new applications, and new questions about how we should use these remarkable tools that bacteria so generously shared with us through the patient work of curious scientists. The molecular scissors that once protected ancient organisms from viral invaders have become instruments of hope for countless people whose lives have been touched by genetic disease, whose communities struggle with food insecurity, whose world faces environmental challenges that seemed insurmountable just decades ago. And perhaps that's the most beautiful part of this whole story, isn't it? That something as small as a bacterium, something that exists in numbers beyond our comprehension in every drop of pond water and handful of soil, held within its microscopic self the key to addressing some of humanity's greatest challenges. It reminds us that we never know where the next breakthrough might come from, what seemingly insignificant organism might harbor the solution to problems we haven't even learned to articulate yet. The CRISPR revolution teaches us patience with discovery, humility before the complexity of living systems, and hope that human ingenuity, guided by wisdom and compassion, can indeed make the world a better place, not through force or domination, but through careful listening to what life itself has learned over billions of years of trial and adaptation. So, as we reach the end of our gentle journey together through the molecular landscapes of genetic possibility, take with you not just knowledge of how CRISPR works or what it might accomplish, but a sense of wonder at the interconnectedness of all living things, the power of human curiosity to unlock nature's secrets, and the profound responsibility that comes with being able to edit the very text of life. The whispers of science will continue long after our conversation ends, carried forward by researchers who work late into the night, by students who ask questions their professors haven't thought of, by dreamers who imagine applications for genetic editing that none of us can envision today. They whisper of a future where genetic diseases become relics of the past, where crops adapt gracefully to whatever climate changes await them, where ecosystems damaged by human activity find new resilience through carefully applied biological solutions. But most of all, they whisper of hope, of the enduring human capacity to learn, to grow, to heal, and to use our growing understanding of the world not for destruction, but for creation, not for division, but for healing, not for the few, but for the flourishing of all life on this remarkable planet we share. Rest well, fellow traveler, and dream of molecular scissors dancing through the night, of guide RNAs finding their perfect matches, of cells repairing themselves with newfound wisdom, and of a tomorrow where the ancient conversation between bacteria and viruses has somehow become a tool for writing a better future for every living thing on Earth. The story continues, and as all the best stories do, with quiet hope and endless possibility.